Okay, thank you everyone for coming back from lunch. I'm sure we'll have a few people come in afterwards because I was having great conversations out there and ignored the bell too. We're into the afternoon of our final day of the symposium, and it's my enormous pleasure to be able to invite Mike Grundy to chair the session on environmental standards, methods and protocols. And you've probably all worked out by now that these topics are very elastic. We have a lot of things that go under them. Thanks.
Thanks, Beryl. Throughout my career, I've been fascinated by the question about the tension between standards and innovation. So I'm actually hoping that we do explore this in this standard session that we're going on now. So here we are, we have, off we go again, we have a potpourri of petite but particularly wonderful things for you to enjoy. And the first talker is Wes.
Okay, good afternoon everyone. It's great to be here. My name's Wes Cooper and I'm very lucky to be presenting the results of my master's research project where I developed a new technique for the spectral calibration of phenocam imagery, which sounds like a rather dry title, but it actually is something I get very excited about. Just a little bit of context, I come from a background of photography, printing and colour management of images and have kind of learnt about the power of spectral data coming from that context and have then applied it into science. So as many of you are aware, phenology is the timing of cyclical biological events, so things like plants flowering, the migration of shorebirds, and as a cyclist, my least favourite, the swooping of magpies in spring. Changes in the timing of phenological events can be an indicator of environmental change, and so it is important that we monitor these events. So phenocams are digital cameras that are mounted in the field to take photos of vegetation at set intervals, and we can process these images and extract indices for monitoring the vegetation. So the initial attempts just used basic webcams that were just mounted outdoors and have progressed to cameras like the one you see here, which is mounted in the US. So the most widely used index is the green chromatic coordinate, or GCC, and that is simply the proportion of green in a RGB image. And phenocams typically use compressed JPEG images. And the colour of the vegetation recorded can change according to the lighting conditions. So the same tree can look a different colour depending whether it's a cloudy or a sunny day. And this is not great. So my research question was, are spectrally corrected raw images better than JPEG images? So to test this, first I verified raw versus JPEG images using ground truth measurements, then analysed the variance over time of the colour data between the two image formats. And then, as an alternative to GCC, I tested hue as an alternative index. So this research was conducted at the Jinyan TERN super site, which I worked out yesterday is the site that is furthest from where we are standing today. It's about 65 kilometres north of Perth in the Swan coastal plain, and it's dominated by Banksias, and it is part of the threatened ecological community of Banksia woodland of the Swan coastal plain. So it's quite a method-intensive project, so I'll try and keep it brief. I used 3 Canon PowerShot cameras, which is just your basic digital point-and-shoot camera, and these were wired up to a central data logger, which triggered them every half an hour between 10 and 2 o'clock. To enable raw capture, I just used the Canon Hack development kit, which is an open source project. It's just a firmware that you load onto an SD card and enables raw capture. A critical piece of information you need to have for this whole technique is the spectral response of the camera. And this is quite difficult to measure. You need a specialist piece of equipment, and despite my best efforts, I could not get hold of it. So I had to develop a technique to estimate that, and that is what those RGB curves that you can see are. So in terms of RAW versus JPEG, so a JPEG is a photo saved off a camera, and it is the finished product, so everything is baked in. So the white balance, noise reduction, lens distortion, a gamma curve. It's basically what you see is what you get. A raw is basically straight off the sensor, so it's linear sensor data, and it needs to be processed, and it is much more flexible. And then for my ground truth measurements, I used an ASD Field Spec 4 spectroradiometer. So for my ground truth verification, I used a metric called delta E 2000, which is basically a measure of colour difference, where 0 means the colours are the same, and 100 means that they're opposite. So I measured 4 different plant species. So what you can see here, the left-hand column is the spectral reflectance of each. The first green column is the ground truth colour, so that is that spectral reflectance using and a colour calculated using a standard illuminant. The second green column is the colour I got from my raw images, and the third green column is the colour from the JPEG images. And as you can see, they're quite different. And I was very happy when I saw these greens. So the mean delta E of 3.49 compared to 21.7 is quite a significant difference. And you can see on each of the green patches, I've listed the green chromatic coordinate and the hue value. I should explain that hue, for those that are not familiar, is essentially a colour wheel. So I'm sure you're all familiar with a colour wheel, and it is measured in the angle as you go around the colour wheel. An interesting anecdote was sample C, Sterlingia latifolia, it actually changes colour throughout the day. And so from my first image at 10 o'clock to the image at 2 o'clock, the same plant would look a different colour. And after seeing this, I actually saw them out in the field, and I would walk around it and it would change. So I think there's something happening with the pigments in the leaves, but that needs further research. So looking at time series data, I looked at an individual flowering event in a plant, a Verticordia nitens, and down the bottom you can see the actual images. So what you can see in the 2 plots here are the comparison between the hue, which is the top chart, and the GCC. And then the solid black line is the raw images, and the dashed black line is the JPEG images. So the hue that is captured from the raw images shows a clear correlation with the flowering event. There is a slight correlation with the hue from the JPEGs, and the GCC from the raw files, but the GCC from the JPEGs really shows little correlation with the flowering event. And you can also see that the GCC from the JPEGs has a lot more variation in the data over time. So, and for each of the time series, there was a significant difference between the raw and the JPEG indices or image formats. So what we can take from this is that spectrally corrected raw images are much closer to ground truth colour than JPEGs. Time series data, hue aligns more closely with observed phenomena than green chromatic coordinate, especially when using spectrally corrected raw images. Hue from raw images provides greater ability to detect subtle phenological events, which is especially important in Australia. The whole spectral calibration technique creates potential for higher inter-site and inter-instrument agreement, which I think can only be a good thing. And a full spectral calibration where every image is calibrated with exact spectral data would need further research. And I just have to thank my supervisor, Caitlin, who couldn't be here today but is online. This project would not have been possible without her, and I'm very grateful. So, thank you.
Good afternoon. I'd like to acknowledge the traditional owners of the land we meet on today, and also the Kaurna people, who are the traditional owners of the Adelaide Plains, where our group is based at Adelaide University. My name's Katie Irvine. I work for TERN in community partnerships and also science communication and engagement. And I acknowledge my co-authors on this paper, Sally O'Neill and Peter Storer. So I'm going to talk to you today about citizen science at TERN, a couple of projects we've been working on, and also the way we are including the standard ecological monitoring protocols for Australia with this citizen science project. So I'd like to acknowledge all of the people here on this slide and also point out that citizen science isn't ever done in silos. It's a large collaborative effort. Particularly, I'd like to mention the citizen scientists, so the group of birders. It's a bird monitoring project that we're working on, and they're led by Peter Storer. Thank you to Beryl and Beck Christensen for played key parts in setting up this project. And we run the project out of the Sanford Ecological Research Facility, or SERF, just here outside of Brisbane. So I'd like to particularly thank Laurel Allen and Marcus Yates, who always make it a pleasure for us to visit their site out there. So I mentioned the standard ecological monitoring protocols. Sally will talk about them in more detail later in this session, but I just wanted to put up this slide so you can understand the context we're working in for the Citizen Science Project. Sally and Ben's team at Adelaide Uni have been working on these protocols for about 2 years now, writing 24 distinct modules for monitoring natural resource management regions across Australia. And so for the Citizen Science Project, we wanted to look at the vertebrate fauna module, and in particular the bird monitoring protocol within that. That protocol is based on BirdLife Australia 2 hectares in 20 minutes survey. And I also should say that as part of these protocols, our tech team in Adelaide are also developing a custom-built app that will allow the NRM regions, so on-site ecologists and consultants, to collect all of the data in a standardised way and feed it through to a central database eventually. So we've been beta testing that up with the citizen scientists too. So we had a kind of a broad question to ask at TERN, which is, can citizen science be used as an effective tool to monitor ecological processes and variables in the long term, and also to expand the temporal and spatial scale that we can cover. Our previous experience with Wild Orchid Watch, which was a citizen science project we ran from the TERN Adelaide group starting about 5 years ago, from that experience, we knew that we could build a custom app and it would be used by the community. So that was a national project. It's still ongoing. And during that time, iNaturalist really came into its own in Australia. And so our data now is delivered to iNaturalist, where that beautiful community of orchid enthusiasts across Australia can share their data and then also provide identifications for each other, and we can host all of the data there. So that's just an aside in terms of citizen science that has been run at TERN. But back to our bird monitoring project, we also were really keen to test these standardised protocols over the last year or so as they were being developed, and our bird watchers up at Sanford have been amazing in giving us really great feedback. So the ambitions for the project, from TERN's point of view, we wanted the project to be long-term monitoring. We wanted to use the standardised protocols for consistent methods. We wanted to collect really tight metadata. And overall, we wanted it to be a streamlined data collection process, and the management and publishing all to happen in a nice flow. For us, for the birdwatchers, these people are volunteering their time. They're expert birders, but they're not on the payroll. So we really wanted the project to be enjoyable and worthwhile, to have these defined methods to make it really clear to everyone exactly what data we were collecting, and to be sustainable for the volunteers over the long term, because it's really important that this, if possible, data collection can go on for as long as possible. And so really what we're looking for, all of us involved in the project, is trusted ecological data, which can be used for research and management and to inform policy and planning. So just like all TERN's open source data. So just quickly, I've already mentioned we were at the QUT SURF facility, which is northwest of Brisbane. We already had 3 soil and vegetation AusPlots monitoring plots set up there. They're the pink squares, and so we overlaid over them the 2 hectare bird monitoring plots. And we had the Sanford Eco Corridors community group, where most of the birders come from. So the project was launched in April 2021. It's been going for over 2 years now. We have 18 surveys that have been collected over 9 individual events. And so that's a quarterly survey effort, and the data collection is still ongoing. And the data has been published. And as far as I know, this is the first time that we've published citizen science data in the TERN data portal. So it's really exciting. That's all publicly available data now. It's worth saying that Peter and the Eco Corridors community group have been collecting bird monitoring data at their Eco Corridor site for 10 years now, so they have an amazing data set of bird data, and that's kind of what we're aiming for here. In terms of future citizen science projects for TERN and the long-term ecological monitoring community, I guess we would think about where are the data gaps and which projects would be suitable for volunteers and community members to collect that data, which citizen science groups, so school groups or undergrad students, online communities of people, for example, the orchid enthusiasts. So who would be looking to partner and what kind of collaborations could we set up? And then from there we can work up a project and have a look at what kind of technical tools we need. For example, apps make citizen science very smooth, and then look at things like developing communication and engagement, education kind of programs. And that's all from me. Thank you.
Hi everyone, my name is Vilim Filipovic and I'm from Federation University currently and also from University of Zagreb in Croatia. So I will talk a bit about European perspective and critical zone observatories, specifically about quantification of water balance. So what is the importance of soil-water balance? We heard few talks and from different presenters here that it's very important to have proper quantification of water balance in terms of monitoring and also in terms of modeling. So it's important for agriculture, for water resources management, ecological balance, water quality, flood and erosion control, etc. And also it's really important to calibrate new models. So when we talk about water balance, we have this simple equation which accounts for precipitation, evapotranspiration, maybe we have irrigation, runoff, and drainage, and we have soil water storage. So that's the basic equation. And of course, it's proper. We need to quantify it properly. But are we missing something? Yes, we are. We are missing some components, and these components are capillary rise. And also, not many people know that it's very important, especially in arid environments, to know about non-rainfall water inputs, which are dew formation, fog, soil water absorption, and soil water absorption vapor. So how can we quantify that? Before, probably you know about the lysimeter system, but now the technology has developed quite a bit. And it's basically a cylinder which you can push into the soil, and you have intact soil core, which is set on a precise scale. And lately, there was also introduction of bottom boundary conditions in which the bottom boundary are replicated from the surrounding soil using the sensors. So you have really high precision. So you have these big soil cores, which can be 1 or 2 tons. And you have high precision, 10 grams of mass, and high temporal resolution, 1 minute. And you can have capillary rise measurement, and also you can estimate non-rainfall water contribution for those systems. So I will present three critical zone observatory sites from Europe. I lead the Superhill in Croatia. It's not in one of the beautiful islands. Unfortunately, it's in continental part, which is close to Canberra climate, so very harsh winters and, and very, very warm summers. So we want to quantify subsurface preferential flow and agrochemical transport. In Germany, they have big SOCAN network of lysimeters for big sites. And they want to have a large climate change experiment in which they displaced the soil cores throughout the Germany, and they look at climate change effects. And in Spain, they want to quantify the interaction between the land and atmosphere. So this is a Superhill site. It's very detailed instrumental hill slope, vineyard hill slope, in which we have more than 100 sensors, water collection, flux collection instruments. It's also quite considerable to notice that when you measure some properties in the soil, and when you measure flux, it's much more relevant to measure flux than if you measure, I don't know, water content or some other properties, then you need to use some model to estimate the drainage. In this sense, when you use weighting lysimeters, you assess it directly. This is just a few examples in which we use and how can we use the data very, very detailed. So we had two weighing lysimeters at the top and the bottom of the hill slope. And what we wanted to notice if there is some subsurface preferential flow. So what is happening, we have precipitation, then we have biopores, and then part of the water is transported preferentially, and then along the hill slope, because we have very compacted horizons, to the bottom of the hills. So we noticed that at top lysimeter we have dominant vertical flux, and at the bottom we have dominant lateral fluxes. So here is just a signature of two. You can see the weight and you can see the rainfall, how it's changing in one season. And you can see on the bottom picture that the tank's system responds differently. So you have the pumps inside of the lysimeters, which pump up the water inside if there is need. So you have capillary rise, or you have a situation in which the system pumps out the water. So we were able to estimate drainage and capillary rise and also subsurface lateral flow. In terms of terrestrial network, we did a measurement on evapotranspiration, and it was noticed there is also a problem that we are not measuring nighttime evapotranspiration. And with lysimeter system, we noticed that this is occurring mostly in wet conditions. And evapotranspiration, nighttime evapotranspiration range between 3 and 9% of daytime annual evapotranspiration, which can be quite noticeable. In terms of precipitation, lysimeters can also be used to capture precipitation very accurately, because you know how much it fell on the ground. So this study showed the differences between different measurement of precipitation, tipping bucket, variable, acoustic, and laser. And you can see these catch ratios were very, very broad compared to the reference point, which was lysimeters. So large underestimation of precipitation, which is actually reaching the ground level. Does it matter? Of course, we did a numerical study in which we were inputting different precipitation and measuring and modeling what will happen. You can see large differences between the drainage, between these two values, and also there is importance of using that instruments or that parameters to capture transport processes in soil as well. And the last, what I want to show, maybe for some people that work in more arid environments, especially now here in Australia. So we notice in Spain that there are nocturnal fluxes which significantly affect water and energy balances. And non-rainfall water provides small but consistent water inputs to our ecosystem. And we noticed that the inputs was around 8% of total atmospheric water. So dew was 20 millimeters in our water balance, so which is quite substantial in that case. And at the end, of course, I don't have that much time, but you can visit the sites and find out more information. I'm sure you know about Terreno, but you can visit also the two other observatories. So myself, Alana, Vedran, and Thomas are involved in Superhill. I would like to acknowledge also John and Daniel from Spain, and also Harry Ferraken and Thomas from Terreno. They are able to provide me with the information and the data which are presented today. Thank you very much.
Good afternoon. I'll take you home to a research case study that we did just here. Last year there was a big flood event in southeast Queensland, and I'll present the work we did at the University of Queensland with Stuart Finn. So in February and March 2022, big floods, large floods in both states. And in Brisbane itself, over 3 days, more than 750 millimeters of rainfall came down, so it broke previous records, so it's a huge amount. And this resulted in significant flood, as you can see on the Brisbane River. Note that this is a very short and rapid flood, so the water rose, and after a day or two, it came down. Okay, so remember that. And these are just some photos to show you how it looked like in the suburb of the university in St. Lucia. And also, you notice all the things that residents put out on the streets, and the army came and took off because everything got wet. Okay. So given the limitations of global and Australian flood products, limitation both which are related to the spatial resolution of these products and their temporal resolution, they are not daily flood products. Our aim was to combine daytime and nighttime optical and radar imagery to map the extent of the flood and its impact. So first we wanted to know, okay, so when was the peak flood in our region of study? So we used the Bureau of Meteorology river gauge data, and we can monitor, you can see on the date max, you can see in which date was the maximum height of the flood. And the low pressure system, it advanced from north to south. So in Brisbane, it was on the February 28th, that was the max height of the floods in this area. And these are just some examples of river gauge heights along the Brisbane River. Note again, okay, it's rising high, and then it's rising fast, and also receding very fast. So it's critical to look at the right time. Okay, more or less. To map the flood extent, we used optical planet imagery from both before the flood and during the peak flood, so February 28, a Capella SAR imagery from the same date, and historical flood extents from the Brisbane City Council, which you can see on the right-hand side in blue, the 1893 flood, 1974, and 2011 floods. We devised a new spectral index, which we call the Normalized Difference Inundation Index, which is a normalized difference index based on the infrared before the flood and the infrared during the flood. And this index, we'll soon see it. Okay, so this is the planet image a bit south of here. This is the Brisbane River, that big snake. So this is before the flood, and this is during the flood. So you can see the, okay. And the index itself, it shows us in blue, I mean, I colored it in blue. We can see the newly flooded areas in blue here, and the red, that these are previous water bodies which are now covered by sediment-laden waters. So it also differentiates nicely between two types of flooded areas. So using a simple threshold, we can map, of course, the extent. This was a bit smaller than the extent of the 2011 flood, which we can see in this cyan. And we were able to produce such a map, when this is after refining, but such a map after just 3 days from the flood event itself. The Brisbane City Council flood extent was only published months later, based on dozens of hydrological models and surveys going to the location and trying to actually measure what was the height of the flood. And these two things are complementary. But again, it's timely response is very important, of course. These are maps showing a bit of a more regional area. So a larger area was mapped. With the Capella SAR imagery, which covered a smaller area, we also wanted to compare that to that result to the product of the optical imagery. So using an adaptive box filter to remove some of the noise. And then again, a threshold applied to map the flooded areas. But as you can see, the areas identified by the SAR imagery in magenta were much smaller than what was actually flooded. So this is probably due to obstructions by buildings and vegetation within this dense urban area. The last thing we wanted to do is also to look at the impact on electricity, on human activity, To that end, we use the night lights from the VIRS NASA sensor. It has a special day and night band, which provides a global nightly night lights data at 500 meters. So we examined all lit grid cells and looked for statistically significant changes in night lights in the period before the flood and after the flood. These are locations where there was a statistically significant change, reduction in night lights. Zooming in, you can see that it corresponds quite well with the actual flooded areas as mapped using a planet in blue. We also compared this with data on electricity loads from Energex. Energex is the regional supply of electricity in southeast Queensland. So these are the polygons or the substations. And again, so the regions that they have are different, of course, from what was actually flooded, but again, spatial correspondence, and over time, these are nightly time series. So the top figure shows nighttime values, nighttime brightness, if it was not cloudy. If it's cloudy, there's no data. The bottom is the actual data from Energex. This is for 3 suburbs in Brisbane. This is for the city of Gympie, north of Brisbane. Notice that we can see not only the impact of the flood, okay, so that there's much lower electricity, but also the recovery. So the recovery takes time, and it differs between areas. And recovery can last much longer after the water has receded. So from space, an area might be not flooded at all. But using night lights, we can tell, okay, is there still something going on? Okay, still people are not having electricity. So key points, importance of real-time mapping. So we need constellation of satellites. We use the Planet Optical and Capella SAR. And I think it's very important for Australia to have its own satellites. So in times of crisis like the Black Summer or flooding, Australia can direct its own satellite to where it's needed and won't be dependable on other countries. In our case, Optical worked better than SAR, but Optical, of course, cannot penetrate clouds. And night lights are a great indicator of human activity. So thank you very much.
Hi, my name is Donna Lewis. I work with the TERN Ecosystem Surveillance Platform, and I will be talking about enhancing the TERN Australia Soil and Herbarium Collection with an emphasis more on the herbarium collection. So ecosystem surveillance has a network of plots along transects in some cases to look at what is changing, what direction is the change and the magnitude of change. Now, within each plot, it's a 1 hectare plot, and we collect a whole range of information, including quantitative data, such as vegetation structure, composition, soil characterisation, as well as a whole bunch of samples and specimens on soil and vegetation. We also have a photo method, an image library, such as the panorama below, and we collect this information using a field app for standardised data collection. We provide many other services, including practical hands-on training and advice, and we've developed a whole range of protocols. So delving a little bit deeper into the samples and specimens, we can categorise these into 4 samples and specimens, if you like. Soil samples that are collected from a 1 metre pit on the plot, including bulk density, soil horizons, as well as metagenomic samples that are collected from 9 subsites across a plot. We also have herbarium specimens, 51,000 currently, and then mainly vascular plants. We have leaf tissue samples that are also stored in silica gel for long-term preservation. Where are these samples and specimens stored? So we now have the TERN Australia Soil and Herbarium Collection, officially opened last year in December, and it was launched by the National Soils Advocate. It's actually situated on the University of Adelaide Wake campus in Herbarium, and we're currently drafting collection management guidelines. As part of this process, we registered the herbarium collection with Index Herbariorum, a global network of herbaria. So here we are, TERN, the TERN collection in Adelaide on the global map, and our code for Index Herbariorum, a unique code, is TERN. Collection management includes the physical collection. So we now have our purpose-built facility. It's climate controlled, has an integrated pest management plan. We are accredited to import and accession the samples. We've factored in expansion of the collection, and the specimens and samples are available to loan for various research projects. We've also followed guidelines for managing Australasian herbarium collections for our herbarium and leaf tissue samples. Collection management also includes the digital collection. So currently we store this information in Postgres. It's a powerful relational database, and it can interact with Specify. So Specify we have set up for our collections. It's a biological collections data management platform. We're part of the Specify Collections Consortium. Australia Herbaria are currently migrating to Specify. So there's so much help and support out there from the consortium and from Herbaria. We can pull information from Postgres, including site information, site location details, and then of course we can pull across our unique identifiers, which are our barcodes that we have on all of our samples. So we have 4 collections set up in Specify, ready to operate. So collection management, looking at Specify 7 in a little bit more detail, we can manage our permit information, our loan applications. We can also manage our exchange program with international interstate herbaria. We have a storage tree, so we can locate the physical samples and specimens in the collection. We can establish routine queries, for ease of staff to access. So just a little bit more on value adding to the herbarium specimen collection. So we've developed vascular plant collecting guidelines. So it all starts, the quality starts in the field, pressing the specimens, making sure they're dried. We can then capture additional information such as habit, phenology, life stage, specimen condition, automating vegetation community descriptions, using controlled vocabularies, and using APIs and services such as the Australian National Species List. So we're using accepted names of Australian taxonomy. Now, once we've collected our specimens in the field, we bring it back into the lab, and we've started a new process of mounting and preserving the specimens. So also have developed guidelines in very final draft. Herbarium specimens are intended for long-term study and storage, so we need to use archival materials free of acids. Otherwise, specimens will degrade and discolour over time. So we use a range of archival material listed here below, and we've also prepared a label including additional information and the TERN branding. Now, the TERN branding is really important because once we start digitising the collection, we have our branding there in the digital image. So we've developed this workflow for anyone to use, anyone as part of the mounting and preservation process, easy to follow. And we also have started digitising the TERN herbarium collection. We've scanned probably a couple of hundred specimens now, so growing. So we've developed these guidelines. Now, digitising plant specimens really started back as part of the Global Plant Initiative to digitise all type specimens globally. It was funded by the Royal Botanic Gardens Kew, and the guidelines were developed to ensure consistency. So the images were high-resolution images, and Australian Herbaria was supported throughout this whole process and provided leaf aptus cameras, and are now delivering it to platforms such as the ALA, the ABH, and GBIF. We've also tapped into learnings and specs from the National Herbarium of New South Wales. So we've used some of their specs as well as following the JSTIL guidelines, which are now captured in our guidelines. We've also developed a workflow for digitising the collection. So really easy, setting up the hardware, preparing the specimens, and then using the software, MyBotany and the Scan Wizard. And of course, following this, now we're capturing all of this new information. We're working with Guru's team and ensuring that our samples and specimens are captured and made available through ecoplots, using controlled vocabularies. All of our images of our specimens and samples are available in eco images, so that work is in the process. We can't forget about the TERN AusPlots R package. There's a version 2 coming soon with many new features. Make sure you check that out. And we're making our information discoverable through international and national platforms using APIs. So EcoPlots has an API that the ALA hits to deliver our occurrence records to the Atlas as a preserved specimen, because we're specimen related. And of course, the ALA being the node for GBIF, it is automatically delivered through to GBIF. And thank you very much. If you have any questions, you can email me. Thank you.
Good afternoon. My name is Arko Lucieer. I'm a professor in remote sensing at the University of Tasmania and head of School of Geography, Planning and Spatial Sciences. I'd like to acknowledge the Palawa people of Lutruwita, Tasmania. And I'd also like to start by acknowledging TERN for long-term support for the drone program, and Purnima Sivanandam, who's done a lot of work on this project, Ben Sparrow and Matt Stenson. So I'd like to give you an overview of progress we've made in recent years in using drones for ecosystem monitoring and mapping. The aim is to establish a long-term monitoring program using drone remote sensing to support ecosystem science. And there's a lot of, drones have been hyped for the last few years. Lots of people are using drones as this sexy technology. But one of the issues is that people just fly with all sorts of settings at different heights with different platforms. And so protocols in this context are actually really important if we are to use the technology for systematic and robust mapping and monitoring. So this project was all about developing protocols for data collection, developing protocols for processing to produce analysis-ready data sets that can be used by the community and that can be used for consistent monitoring. That also then includes training of TERN users and making this technology operational, so it moves out of the research space into an operational domain. So a brief overview of the hardware. So we selected the DJI Matrice platform. It's robust. It can handle dust and a little bit of rain. Has obstacle avoidance and can fly for a reasonably long time. And all of these things are important to make the flying robust, but also to cover a reasonably large area. So we can easily cover sort of 200 by 200 meters in great detail and scale that to 400 by 400 meters for calibration validation purposes. We capture imagery at 1 centimeter resolution, and I'll tell you a bit more about the accuracy in a minute. We also fly this 10-band multispectral camera, where the spectral bands in the visible and near infrared are partly aligned with a Landsat and Sentinel, so that we can collect that non-visible data as well. And one thing that's really exciting is a recent development in LiDAR or laser scanners with a relatively low-cost laser scanner that's compatible with this platform that allows us to capture 3D point clouds at close to 1,000 points per square meter. And it's really exciting in terms of being able to measure ecosystem structure, which is notoriously difficult to do both in the field as well as from space. Second part of the talk looks at the protocols. So protocols often have this sort of boring connotation, but they're extremely important, and we've seen lots of fantastic examples already today and in this conference. So protocols involve things like checking the equipment, setting up the drone, determining flying height, flight speed, potentially adjusted to the ecosystem you're observing. If you're flying over rangeland, you probably have to fly a little bit lower compared to tall forests. It involves things like camera settings, the white balance, some of the settings in the earlier talk. Do we capture JPEG or RAW? What are the shutter speeds? All of those things are important for robust and repeatable data collection. Then post-flight, how do you back it up? How do you label your data sets? How is it uploaded? How is it made available? Back in the office, we process the data. So we go from the raw images or the raw point clouds to analysis-ready products. So again, there's protocols there to use a standardized workflow to ensure that we always do the same thing. And this is both in the image space as well as the LiDAR space. So I suppose the most important thing, the data products, what do they look like? So in the TERN project the last couple of years, we had an opportunity to do an accuracy assessment to determine the absolute accuracy of the analysis-ready data that comes out of it. And one important criteria that we tested was not using ground control points, because physically putting out markers in the fields is very time-consuming. And if we want this process to be fast, we don't want to lay out ground control points. So with this RTK GNSS system on the drone, we developed scripts to co-register the RGB and multispectral data. And so just based on a local base station, we can achieve 2 to 3 centimeter horizontal accuracy at the operational flying heights, and about 10 centimeters vertical. So I was quite amazed and surprised to see that after several tests. And for me, it sort of gave me confidence in using the data for change detection. So this is just a snapshot of a site in Tasmania. You can see some animal trails and branches in the high-res RGB imagery, and then overlaid on top is the multispectral imagery. So there's no photoshopping or no PowerPointing here. I haven't shifted the image. So you can see there's almost perfect overlap between these images, and they're from different sensors stacked on top of each other. Now, we then broadened the trial to a range or variety of ecosystems. Last year in May, we teamed up with the surveillance team, Ben Sparrow and his team at Kelpram, and also sort of tested some of these protocols. Can they be used and easily adopted by people or a team who is relatively inexperienced in drone operations, but very experienced in field operations? And so we selected 18 Ausplot sites, flew all these sites with multiple sensors. And here are some results. So this is a river gum site. Again, multispectral imagery. So the colors you're seeing are false colors. They represent reflectance in near infrared. And then in the back end, we developed several Python scripts to automate things like image segmentation and a derivation of vegetation indices like NDVI and so on. So that very quickly from the analysis-ready multispectral mosaics, we can move to derivative products that are typically used in remote sensing. We also tested the LiDAR sensor. So here's some snapshots of that same site in a 3D point cloud. The points are colored by RGB colors as observed in the imagery, but you can see the sort of richness in the structure in this data. We developed an automated processing workflow based on R and LIDAR that allows us to identify ground points, produce a digital terrain model, digital surface model, canopy height model, vertical profiles, and again, these are all sort of sped out at the back end and delivered as spatial data products. We started experimenting with individual tree detection and segmentation, and it's still notoriously difficult in natural and complex vegetation communities. And this is an example of ray cloud tools developed by CSIRO, where we're testing the accuracy of individual tree segmentation. This is ongoing work. It's not robust yet, and this is certainly, I think, where collaboration with the artificial intelligence community is really important, because this is where progress really needs to be made to extract the ecological building blocks from the raw data. So some additional current initiatives. So this is work that I've reported on from the last couple of years, developed under TERN Landscapes and in collaboration with TERN Surveillance. In addition, we're also focusing on hyperspectral data collection. So we have a bigger drone with hyperspectral push broom sensors in the visible near-infrared and shortwave infrared that we can mount both on the drone as well as a light aircraft. So depending on the size of the area, the scale we need to capture, we can sort of choose the platform. This is some material from a postdoc of mine, Emiliano Cimoli, so showing that we can extract traits from the hyperspectral data. So we're looking at things like chlorophyll content, carotenoids, anthocyanins, so mapping pigment concentrations at that sort of 4 to 6 centimeter spatial resolution level. And that allows us to look at these physiological traits across the landscape and also look at the variability within crowns. So in the next phase, we're really keen to deploy this system across super sites and dedicated CalVal sites for satellite calibration and validation. Then there's Lara's work. So Lara spoke this morning, focusing on AI tools and AI like deep learning techniques, convolutional neural networks, to identify ecological building blocks from the raw data. So can we use these techniques to map effectively shrubs and photosynthetic, non-photosynthetic vegetation and bare soil, and then produce these as spatial data layers. So where is this going? So we'd love to scale up this effort and get dedicated teams in the field that to collect data across the country, the OS plots, but also dedicated sites for calibration validation work. We'd like to extend the protocols to tall and complex forests and optimize them for those environments. And also develop protocols for the sort of high-end sensors, the hyperspectral sensors and LiDAR data collection. We're really keen for TERN to play a big role in national and international calibration validation and work with organizations like Geoscience Australia, Digital Earth Australia, to really contribute to the national CalVal effort. And this is where the TERN community, I think, has a say over, well, what sites do we prioritize, where do we deploy field teams, and what are the key ecosystems to start with. And then furthermore, finally, we're looking at those AI tools to extract higher level products from the analysis-ready data sets. So these are some long links that I'm happy to share separately, but when you search for drone on the TERN data portal, you can find data that you can play with and that's now freely available and that we're planning to expand in the coming years. And then finally, again, I'd like to thank my team and TERN for their fantastic support over the years. So thanks very much.
Good afternoon, everyone. Thank you for sticking around. Thanks, Arko. I am going to continue with the theme of protocols are not boring. So yes, I'm Sally O'Neill, the Operations Manager at TERN Ecosystem Surveillance based at the University of Adelaide. Today I'm presenting on behalf of Ben Sparrow, who's been mentioned a few times before, and Andrew Totmakoff, who's not here today. Andrew's in charge of the app this project is around, and also Guru, who takes the data from the app and sends it to a database and does all the magical stuff. And Ashley Leedman and Sandra Walpole as well from the federal government, from the Department of Climate Change, Environment, Energy and Water. A special mention also to Janice Goodwin and the team at NRM Regions Australia as well. So yes, my title, Ecological Monitoring System Australia, or EMSA. You're going to get that thrown at you a few times during this talk. Fortunately, there's not too many other acronyms. And I would also like to acknowledge the traditional owners of the land on which we're here today. I also want to go on theme with this actual title of this project and acknowledge also the traditional land practices that the traditional owners of this country have been operating and conducting for thousands of years, and acknowledging the need for them to be able to continue to do so. Natural resource management in Australia, for the scientists in the room, a quick recap. NRM in Australia is predominantly about the people. Yes, we're all working for the greater good. We've got some really good themes that we're all trying to contribute to do, but it's about these guys up on here. They've all got smiley, happy faces. If you're online and you're an NRM person and that is your photo online, please email me for your prize, because I did pull for all these photos from Facebook. So NRM in Australia isn't just about planting trees, controlling pests, and weed control. They actually do a lot of monitoring. Here, just in these photos, for example, we have the monitoring and evaluation network, some really good fire response projects. The guy there with the big pole is actually looking at monitoring the damage of camels, herbivory damage, obviously. We're doing some pitfall trapping. The girls in the bottom corner are actually talking about a project which is spotlighting at night time. And of course, bird surveys. So these people do ecological field monitoring. Unfortunately, it means that every region across Australia is doing it slightly differently. So it's really difficult to pull data together and sort of identify what's been happening across the country. So this project is about the federal government's the program has been called the Regional Land Partnerships. It's in the last 5 years, that's $550 million worth of on-ground projects happening across Australia in our 56 NRM regions. Those projects have now just come to an end, and everyone is frantically developing their projects that will roll out for the next 5 years under the National Land Care Program. It's important to acknowledge that these are big projects. These aren't individual farm level projects. They're big regional projects with some amazing outcomes. They're trying to work towards recovery of threatened plants and species, plants and animals, threatened ecological communities, also rammed up, and wetland protection, and some agricultural land stuff as well. You will know that I'm from TERN. TERN stands for Terrestrial Ecosystem Research, and the wetland side isn't our strength, so it's something that we'll hopefully get to in the future. So as I mentioned before, every region across Australia, we did a scoping study and identified that whilst they're doing some amazing stuff, most projects have actually got their bespoke methods. They've sort of grabbed a little bit of this method and a little bit of that method and pulled it together, and in some cases they've added a few extra fields, which means that extrapolating across the country and comparing data sets isn't really possible. I've thrown here some examples of the documented survey guidelines that are around. There are literally hundreds. We did a big research into identifying everything that was used. So just acknowledging that there isn't one consistent method, and that's what we're aiming towards now. I was hoping that somebody else would use this. I know Matt Stenson used this funnel picture yesterday very briefly, just identifying that TERN works at 3 levels. Of course, there's the landscapes group that we've had from quite a fair bit, and Katie, Donna and I are all from that middle group. We're the on-ground people doing the on-ground plot stuff that Donna just talked to you about, so don't bother talking about that too much. So yeah, the federal government came to TERN for our expertise on that on-ground stuff and our expertise around standardised methods, protocols and collections, which Donna just talked about, and the data infrastructure, which is the group that Guru talked about. You may recognise this manual in that bottom corner. It's been around since 2012. It's the original AusPlots field manual. It's only 60 pages or so. We've taken what we've learnt from this project from the AusPlots TERN surveillance program with 1,000 plots across Australia for the last 10 years, and that's when the Australian government came to TERN and said, right, we need your help sorting out this NRM monitoring for our project of natural resource management. So the work that Donna just talked about, basically we have these modules. So plot selection and layout is where do we establish a plot? How do we accurately locate the plot? We describe the plot. Donna talked about the soils work that we do, the photo points, floristics and plant vouchering was her favourites. We also do cover measures across the plot as well. So taking the work of TERN for the last 10 years, this was our basis that we already knew, but we did some work for the Australian government to now expand. So now this data is now all available, in TERN AusPlots and the TERN Discovery Portal that has been mentioned quite a few times. So now we're extending it. We now have 24 modules. There's a lot. There's a lot of data that's about to be coming in. They're all very exciting modules, but as you can see, it's just the little wetlands, riparian and coastal stuff that is missing, but we feel that we've captured all the data requirements for NRM monitoring in Australia for the time being. Each module, each of those little pictures is actually, imagine it as a button on an app. There's a lot there. 24 modules is a huge, we bit off a lot, but we're coping really well. So from a modelling perspective, or someone who wants to analyse data in the future, be warned that in the next couple of years there is a lot of data coming, and we'll be expecting people to start playing with it pretty soon to understand the capabilities and take it to the next step. So each module is an instruction book, is part of an app, and is a data product. The document itself provides a rationale for why the ecological variables were considered important. We talk about what the literature and methods already established that we've considered. We identify prerequisites, personnel requirements and equipment stuff, so the on-ground stuff that the field ecologists need to know. Our instruction section is very clear step-by-step procedures with some additional guidelines which are essentially hot tips and tricks. And then we talk about the types of data and samples that could be used in the future. So the Ecological Monitoring System Australia will establish best practice monitoring protocols targeted to specific project needs, improve the understanding and measurement of the effectiveness of NRM actions, strengthen the evidence used to assess the achievement of these outcomes, and facilitate better access to and reuse of ecological survey data to lead towards policy and decisions. In considering how we came to all these things, I just wanted to say that we did consider all the little nitty-gritty requirements. We wanted to be built on collected measures, so the quantitative data stuff, not just the qualitative observations. And we needed to consider that these NRM people are stretched already. We couldn't have super onerous techniques that would take them days and days and days to collect data. We needed a few different flexibility approaches to be included. It needed to be practical. We needed to be using equipment that they could run down to Bunnings and sort of construct themselves, not thousands of dollars worth of equipment. And it needed to be suitable for on-ground staff, on-ground ecologists, not scientists. We did have a comment early on in the project that they thought that these protocols were going to be too onerous. They thought that you had to be a full-blown scientist to be able to collect the data, and not at all. It's a really good project. They're great instructions to read, not tooting my own horn, but I think they're really good. And it's definitely designed for on-ground field ecologists with a few years' worth of experience, but it's also identifying that you do need skills. There are plants you have to ID, although we do recommend herbarium determinations. You do record scats and tracks and all that sort of stuff. So in the future, EMSA is all about the NRM project at the moment, but we have had a lot of other areas show a lot of interest, anything from managing our national reserve system, so Parks Australia may implement things like this in the future, biodiversity markets obviously, any sort of land management, so both defence land, forestry land have also reached out as well. In the next few months we'll be designing our training programs, designed specifically for the NRM projects to commence, and collecting our learning and on-ground management actions and research outputs to guide improved decision-making is what this is all about, and that's what we'll be focusing on from now on. So in summary, EMSA is 24 modules. It's a field collection app, which is called Monitor. There's our logo of the lizard. It's a standardised vocabulary using the new developing Australian Biodiversity Information Standards called ABIS, and it's delivered to the Australian Government's Biodiversity Data Repository, the BDR, with the help of TERN. To acknowledge my protocols team, they're all the people picked there in the photos. They're a great team to work with. More information and the PDF documents of all of our instruction manuals are at the EMSA website, and stay tuned because the app should be out very soon. That's all.
Hello, I would like to start by acknowledge the traditional owner of the land we stand today, and that's the Turrbal people, and pay respect to their elders, past and present. Now, my talk is going to be about a new way to assess habitat condition for biodiversity conservation and restoration. Now, to start with, what is habitat condition? Now, a very broad definition would be the ability of an ecosystem to support biodiversity. But depending on the specific approach that one takes and the indicators that one looks, the definition of habitat condition could change actually quite a lot. So here is a summary of the approaches that are widely used. So the first column, what I have are the drivers, which are actually, what habitat condition is about. So for example, if it says soil and vegetation, then, condition score is about the condition of soil and vegetation. Second column is the commonly used example. And then I listed their strengths and weaknesses. So as you can see, I generally look for 5 categories. Now, first is whether the good condition is achievable. Second, whether we can assess the predicted score with actual observation. Third is whether this assessment framework will work on different spatial temporal scales. And the fourth is whether the predicted score is based on actual observation of the ecosystem rather than indirect factors. And then last one will be whether this approach will work on large and small scales and with reasonable cost. Okay, so let me just briefly explain what's happening here. In this table, we can see none of these existing approaches is good in all aspects. Some common problems, including, say, this soil and vegetation-based approach, the top one, is too costly. So it's not possible to do that everywhere, anytime. And then we have other approaches that might not be working on fine and coarse spatial scales. We also have approaches that may not be based on observation. So I would like to specifically mention this Biodiversity Intactness Index, BII. So it's a modeled product based on a number of environmental factors and human activities. So there's high human activities and where there's no land use with heavy modification, then this score goes down. So as you can see, it's not a direct measurement of what's happening in the ecosystem, and it has this assumption that human activities will necessarily result in degradation in habitat condition, which I don't think is correct. Now, the framework I propose here is fairly simple in terms of logic. Once we have a target site, we can then look at the environmental layer of the interested area and then select the sites that with known condition that is in similar environmental range as the target site. And because we have the reference sites, we can work out a probability of a site of being in good condition, and build a relationship between the probability and the actual observed habitat condition. Now with that, we can then predict a habitat condition score for a particular site. Okay, I'll go through this framework with a case study of Australia. the dataset I'm choosing is half plot, which is plot level native vegetation species fraction. And then the predictor I'm using are from Lancet. I have NDVI, albedo, and delta 15N, as well as their variation. Now, NDVI albedo probably makes sense directly. This delta 15N value is something I developed as a predictor of a nitrogen cycle, which kind of indicates what kind of vegetation there are in the ecosystem. So the map on the right-hand side is an example. I'm showing the cross is the target site. The blue points are the reference sites with bad conditions. The green are the reference sites with good conditions. Now, with that information of good and bad reference sites, we can then construct a distribution of a particular predictor. So the example here is, this delta 15N value, and if we look at the distribution of this delta 15N of all the good sites, we have this blue distribution, and then for the bad sites, we have this red distribution. And it's fairly easily for one to say that, if a site has a delta 15N value of say -1, then the chance of its being good is really high. Versus, see if we look at NDVI, in this particular case, we can't really easily separate the good and bad sites just based on NDVI. Now, the good thing about this approach is now we have converted a particular predictor of a site to the chance of the site being good. Now, if we plot the probability of the chance of the site being good condition against the actual habitat condition score, we can then see this logistic regression between the two. So we allow this logistic regression to change over time and from site to site. Now, with the relationship, we can then convert any the indicator value to a condition score value, right? So that's what we did here. So we converted the albedo, the annual median albedo value, to a condition score for 1,000 sites with known conditions. So the x-axis shows the observed condition, the y-axis is the predicted condition. The observed conditions are just 0 and 1s, so the spread is actually added noise. The number, as you can see, those are not just dots, they are numbers. The number shows how many predictors successfully separated the good and bad sites. So if it's say really high, then it's good. If it's 0, then it's bad. So we can say there's not many zeros, and the error is pretty high for this particular predictor. But I'm just going to explain how we are going to deal with that. Okay, now if we do the prediction with all predictors, we will end up have a number of different habitat condition scores, and then depending on the uncertainty of the prediction scores and their error, we can then weight the predicted score from each predictor. So if we then sum up the weighted predicted score of all predictors, we end up have this finalized final predicted score. And then, as you can see, the error is much lower. And also, if we want to look at R² value, it's actually quite good. So with this approach, I'm already making initial maps. Apparently, there's quite a few things that we are going to change. So these are not final maps, but the plan is to make annual habitat condition maps from 1984 and onwards. The example shown is the southern tableland, kind of north to where I work. So that's the Google Earth map compared to the predicted score, but it's at a 1 kilometer resolution just for demonstration purposes. Now, here is the reason why I like this approach. It's extremely flexible. It doesn't really care what you throw at it as predictors. It's also very fast in terms of the data that is processed and the output it can give. The whole process is relatively easy to understand. It's transparent. And the score, the final score, well, at least in my example, it's very easy to understand what the score means. And also, it's a fairly consistent way of getting habitat condition. Although I'm using plot-based data, which I think is the highest quality data, we can easily replace the plot-based data with any other land use map. And we can also use any ground-based or other remote sensing predictors as the user like. And the key thing is because this is such a low cost and low demand approach, we actually encourage very fine scale restoration that were previously difficult to monitor, and then we are not really able to encourage. Running out of time, but if you're interested in the Delta 15 map, it's going to be available, and I'm happy to talk about it in more detail in the QA section. Or you can feel free to get in touch with me via email. Thanks so much for your attention. That's it.
Gosh, it is bright up here. Hi, I'm Maxine Newlands. I'm from the Australian Academy of Science, and I'm a late addition, if you like, which is why it's not on the programme. Today, I am here to talk about a project that we ran on behalf of DQ. The Academy were asked, well, the Minister was basically wanting to look at the reef and the future of the reef, particularly, and that's the Great Barrier Reef, I should be clear. And so, in particular, sort of going forward in light of climate change and lots of local stresses and lots of components which are impacting on the reef. So the Academy were asked by DQ, as I said, in order to run a project which would identify what were the key issues and key concerns and areas of focus going forward over the next 5, 10 and further years. So on this, I was hoping on this presentation to actually give you an overview of the findings of the project, but it is still embargoed. So I'm focusing on, unfortunately, it's a bit like the end of the TV series. I won't be able to give you what the plot twist at the end is, but I can tell you how we got there, and then it cuts off at that point. This is going to be very quick. First of all, obviously, I'd like to acknowledge the country. We are Narrabri people down at the Academy in Canberra. I'm actually work remotely and based in Wulgarugaba and Bindal country, which is also known as the Townsville area in North Queensland. And very, very briefly, the aims of this project was to develop an understanding of the impacts of climate change on the Great Barrier Reef, to bring the best minds together that the country Australia, Team Australia, if you like, could offer, and to establish what would be the way to go forward and how would that help decision making, to develop potentially models that could provide scenarios and situations in which best information and best data could be made, and look to see where effectively Australian government and Australia's science community should be prioritising their focus going forward. So very quickly, by the numbers, there were 3 roundtables that were held at the Shine Dome down in Canberra. and they were over a period of 3 months. They were very, very quick. Each one had approximately 25 to 30 scientists that were invited through a nomination process. We approached a series of reef-related institutions, businesses, organisations, NGOs, regulators, the whole sort of gamut, and we asked them to nominate people they felt were appropriate for the theme of the roundtable. Altogether, we had 86 participants, and that was from 24 reef organisations, which totaled 117 fields of expertise, although we did feel that there was a lot of focus on coral and fish, but that was fine. That was in the first one. We actually gathered 9 different lines of evidence. We wanted to cover every single, look under the sort of doona. And this was, so the main ways we did this, and I'll talk to you very briefly about these. We identified a climate scenario. We used a scenario system following the Sutherland method. That's where you identify a problem and you use the scenario as a way to stimulate conversation. We then sent out a series of pre-surveys before the roundtable, so 3 surveys went out. The reason we did that was that the people that were in the round table, we wanted to, put it bluntly, we didn't want it to be, my name is such and such and I'm here for this. We wanted to make sure that we got the most out of those people in that time that we had. And also the survey helped us focus what the roundtable discussions would be on. So we could see, using the scenarios, what was the process. We then used a further in roundtable process of using Slido, the software tool, which is a way also to stimulate conversation. And then the whole team from the science policy, we had a series of notes, so field notes, if you like, we were taking. Each roundtable was recorded, but it was purely recorded to double check, because in parallel to this process, ANU were running a study on how this worked, because it was a first, and from that they're going to be publishing, along with the Academy staff, papers on the methodology. And that all culminated down into, can you believe it, a 40-page report, which is, no one's going to read the whole thing unless you're in it, but the 2-pager will be out fairly soon, fingers crossed. Okay. So the timeline, as I said, it started on the 17th of March. That was our first roundtable, and the final report, which the purpose of this whole report was to provide advice to the independent expert panel, which is made up of a series of reef experts. The independent expert panel then take the Academy's report along with their report, and they then provide advice to the Minister. This is concurrent, and that's Minister for the Environment, Tanya Plebership, and also obviously the Queensland Minister will be getting the same advice. And this was in concurrent to 2 other major reports. So effectively the ministers and the decision makers have 3 high-level, the best minds in Australia reports for them to make those decisions. And the Academy's role was key in that. Okay, I've already had the 3 minutes, so I really want to get to the point of this talk. So the climate scenarios were done. We ran through what would happen to the reef in the short term and mid-term, what would happen between 2040 and 2060 under a low emission scenario, and what would happen under a high emission scenario in the same timeline. One of the key things, and this is really the point of this talk, is the key thing from this process was we understood it was it's fundamental that we had to work with traditional owners, Aboriginal and Torres Strait Islander peoples. The reef is on sea country. It is sea country, and sea country, if you're not familiar, runs from the starts of the rivers all the way out to the ocean. So through our process, we had 3 chairs. Each chair had a different speciality for each one of the round tables, but at every single one, we had lovely Chrissy Grant, who is an elder from Torres Strait and Aboriginal heritage. And Chrissy, if you're familiar with her work, she does a lot in the international space. She works with the United Nations. She also works with ICUM. At every stage in those little dots where I said the lines of evidence, we consulted with the chairs, and we also consulted with Chrissy Chrissy, and we also made sure that we, under Chrissy's advice and guidance, as well as the other chairs, we invited a range of Indigenous scholars as well. And the reason we did this was because we know, and this is just the stats, we know that obviously if you're doing anything on country, you really have to work and use things like the free, prior and informed consent process. But also this is our reef, it's everyone's reef, and so we really, the purpose was to make sure that we were not exclusionary, that it was a completely co-designed process. So just some very quick, I'm going to do this really fast, just to give you an idea of the round tables. We had the attendance by mode. We had in person, we did and hybrid, so a bit like we've got now. We did so well on the first round table, it was hard to convince people to come to Canberra for 2 and 3, but they did, and you can see the gender balance there as well. As I said, there were a lot of nominees and we were selected. The chairs helped select the participants in order to make sure there was no bias, but also to make sure we had the right people in the room. And this is a picture. You can see there the screen. I won't use the clicker, but that's the online people, and that's the in the room in the Shine Dome on the left-hand side, and we had the screen. So altogether, we also, around the edge of the room, had observers from government, from different government departments, and different reef organisations. So we hopefully got the broad range. These were the different participating organisations. And the multiple lines of evidence. But I think the take-home message I'd like to give you, that we actually found it so enriching having the traditional knowledge as part of the process. There were lots of things that weren't, not to say they were not considered, but things that were brought to our attention and brought to the attention of the room that will then, we've heavily informed what happened in the report and our advice to the Minister. And I'll pause it there. Oh, I'll leave it that one, don't worry about that. And I'd just say, there's the report, there's the front page, tantalising taster. And I just want to say thanks as well to everyone, the fellows, the team at the Academy, and of course the chairs and the participants. Thank you. Might as well stay up here.
Well, I think you'll agree, or you should agree, that that was a very rich set of discussions. I don't know that any of them explored the tension that I wanted them to explore, but I didn't really expect them to do that. We only have 15 minutes, so what I propose to do is to ask a question of each of them, and I'd like each of you to answer it briefly, but brilliantly and engrossingly, please. So if you could please move to that. There are more questions on here. You can all see them, and you can talk to the people who've asked the questions, please. So let's start with Wes. Quite a long time ago now, you no doubt remember that Wes showed you what you can do with an ordinary camera if you're clever. Okay, so Quez, the question here, Wes, is can you see the same capability being used with imagery from drone satellites and other images, and I'll throw into that. RAW isn't RAW and sensors aren't sensors, so surely it matters which camera you choose.
Yes, well, there's a lot of things to unpack there. So drones, yes, definitely. You can apply the same principles. Many drones shoot RAW. You could theoretically attach like a spectrometer to the top of the drone, and it could capture the incoming illumination at the time you take the picture. And if you've got the spectral response, then that would be like a dream scenario. Satellite imagery, I don't know for sure. It seems to, I guess it has to deal with aerosols and atmospheric effects and a lot of things like that. In terms of different camera models, so the beauty of this is if you have the spectral response of the different camera models, you're essentially calibrating them. And so once you apply that, you're going to get consistent colour across models. There is limitations because different cameras have different gamut capabilities, but it's a pretty good solution. So.
Thanks, Wes. Engrossing and brilliant and short. So, Katie, there are quite a few questions here for you, but in some sense they all come to the point of how do you expand what you're doing? What's next, etc. So let's. They're asking questions like, will this integrate with pre-existing bird data that other people are doing? Can this contribute to other modules? Can we get this methodologies and protocols more broadly? So I guess the questions add up to how do you tend to expand from here to take over the world?
That might be a question for Beryl and Ben. The question about bird life is, yes, the data does go there, and that's thanks to Peter Storer, who runs the bird surveys. He uses eBird, which I think a lot of birders in Queensland in particular use, probably across Australia and certainly around the world. And that data does feed, as far as I understand, into BirdLife and then across into GBIF. In terms of other citizen science projects, I guess, as I said in the talk, it really depends where the data gaps are. And if there are data gaps that align well with volunteer collected data, then the world's your oyster and just needs to be resourced. And there are definitely lots of people out there who would really like to get involved in the kind of work that people here do. And citizen science is the perfect vehicle for that. It just needs to be well supported and resourced so that we don't leave volunteers out there on their own. But
Thanks.
Definitely some great data can be collected.
Thanks, Katie. Now, you all, like me, have been excited to see the talk about lysimeters. No real soil scientist doesn't love a lysimeter. We've got a question here around whether or not you're managing sensor drift, and I'll take the prerogative to add in a question about, are you really assuming no lateral flow, or how are you dealing with flow laterally above the bottom of the lysimeter?
Thanks. Very happy that you are excited, and everybody, about the lysimeters, as I do. So the question about the sensors. So you are basically having the system which is confined profile, and the sensors, you have quite a lot of sensors in terms of soil measurements, so water content, water potential, etc. You also have the sensors or suction cups for taking up the solutes. So the sensors are basically behaving like they would in the soil. I think the question was more oriented towards, as you said, bottom boundary condition. And the bottom boundary condition is challenging indeed, because you have the data on 1 minute basis. And especially in Terreno network, you have lysimeters which are on wheat rye type of agriculture. So you have planted wheat, and there is a wind problem. So you have change of weight during the wind. And there are quite a lot of filters, and there are team in Uli doing the filtering of the data and also trying to somehow manually flag the data which are not good eventually, because there is quite a lot of information. So you have basically two levels of information at the top boundary. So if the weight is increasing, you need to establish whether that's some kind of precipitation, or is it capillary rise from the bottom. That's the two cases. And the lysimeter, what was your question about the bottom boundary? The lysimeter is confined, so we are known when the pumping system is initiated, when the system is pumping water inside the lysimeter, so we know that there is increasing moisture around the field. And when it's decreasing, when drainage is pumping out, then we know that is drainage. So we know quite well. And in terms of side flow, there are quite a lot of technicality around that, but it's prevented for sure.
It's prevented but not measured. We'll come back.
Okay.
We'll come back to that. Noam, I've added to the very small number of acronyms that are used in remote sensing, you'll observe. We now have something called the NDII. You suggested that we should have our own satellite doing this sort of thing, which sounds very sensible to me. That means to me that we have the choice for choosing our sensor. What sensor should we choose to do this?
Of course, the sensors vary depending on the application. I think that at least, so a few bands in the visible and near infrared, but what's important is the temporal resolution to have, I think, daily. So that would require a small constellation of satellites focusing on Australia. So they don't have to cover the entire world. They can, so that we pay back. We contribute also to the international community of remote sensing. So we're not just imaging Australia, but they can be focused just on Australia mainly, and to have the agility to side view. So if they have an event somewhere, the satellite can look to the side. Maybe it's a different geometry, but we'll have quicker views on that specific area.
Thanks, Noam. We're going to have to keep people being brief and more brilliant. Donna, I'm excited to see collections of soils and other things as well, I guess. What are the connections that you have to the state herbaria and soil archives? How are you managing those connections?
So I can probably only speak, and I'll keep it brief. And as you mentioned, Mike, with the herbaria, we actually have a presence on the Council Heads of Australasian Herbaria when they meet quarterly. We also have involvement now with the managers of Australasian herbarium collections, so following Australasian standards for collection management and preservation of the collections. And we also now have a presence on the HISCOM informatics group, which is all around the delivery of the data, because TERN now has, preserved specimens. We're not just occurrence records that we collect on our 1 hectare plots. We are actually specimen based. So it's quite unique that we actually collect samples of soils and specimens of plants on these monitoring plots. I can probably present about the soils at next year's conference and answer that question.
Good on you, Donna. I'll come. Thank you. So, Sally, I think we were all exhausted watching your presentation, and the ambition of it is extraordinary. There are a number of questions here, and I'll ask you to catch up with people about around those, but perhaps you could somebody has actually suggested that it's not broad enough and you should incorporate Indigenous knowledge as well. Are you planning to do that?
Absolutely, and to keep it simple, we decided quite a while ago that if we're going to do Indigenous knowledge collection, we're going to do it well, we're going to do it properly and considered, so we'll take our time and get it right with co-design. So that's a project that will hopefully start soon.
Thank you, Sally. Arko, there are, again, many questions here for you. Drones do get people excited, it seems. And I've got actually a number of questions I'd like to add on, but that would be a bit rude of me here because there have been many others for you to talk about. But I think probably the question around the connection between whether or not you can get to really tricky things like fauna, etc. with your drones. I think there are lots of other questions like geophysics. I'll catch up with you afterwards. But what about fauna?
Well, there's one question that's not really my domain, but there is work on fauna and detection of fauna in drone imagery, certainly in the thermal space. So there's a PhD student at UTES working on this topic. So there are absolutely opportunities for automated counts. One of my students is looking at shearwaters and shearwater nests and chicks in nests and counting hot nests. And so there's absolutely work in that space that's pretty exciting. It leverages the sort of unique viewpoint of drones and the high resolution.
Thank you, Arko. And Jinyan Yang, you told us a picture which is enticing. How confident are you that it works as well in quite different ecotypes, such as, for example, savanna or grasslands?
That's something that we are looking at the moment. From the initial evaluations that I have done, we are reasonably happy with the result. So if you want some numbers, I think the on average, it has an accuracy of around 76%. The average error would be around 0.2. So that's relatively consistent across regions. That being said, so my current approach relies heavily on ground-based plot level data. So when there's not enough reference size for me to actually compare the target size around, we actually have really high uncertainty in the prediction. So if anyone has any better idea of what's out there, I would happily, listen to. Well, let's have this conversation.
Well, I'll leave that up to the community, Jinyan Yang. And finally, Maxine, the add-on at the end, a fairly impressive add-on, I have to say. She assembled all the best minds in the country, apparently. I applaud the use of scenarios. I think scenarios is something that we should be exploring much more. Spent quite a bit of my last part of my career doing it. However, how confident are you that politicians, or perhaps I should say an adversarial political system in this country, is able to cope with the use of scenarios?
Well, I'm going to, the Academy is an independent body, and everything it presents is on evidence-based. So what people do with it is their choice. However, saying that, since doing the scenarios and in other studies, we're actually doing, looking at that in another case for the UNEP for a project on horizon scanning and storytelling. Like you say, it's getting a lot of interest and a lot of traction. And I do think it's something that will, over time, become more and more common.
All right. Again, I think you have to admit you've had a very good afternoon, and would you please thank the speakers? You have to get down now, apparently.
Thank you, Mike. So based on the fact that I didn't get a cup of tea until lunch had actually finished, we're voting you all a little longer at afternoon tea. Can you please be back here at 20 to 4? Thank you, not 3:30. We'll see you soon. And do come back, it's going to be great.